Hello, my name is Nishan Shah, and today I will be talking about the Zika virus. Until recently, the Zika virus was virtually unheard of. However, links to birth malformations and a prediction that 4 million people could be infected this year have led to World Health Organization declaring it an international public health emergency. I had the opportunity to visit Zika virus for my microbiology project. Zika virus is a member of the flavivirus family. It is similar and is related to other pathogenic vector-borne flaviviruses such as dengue, yellow fever, chikungunya, and West Nile virus. Like its relatives, Zika virus is transmitted to humans primarily through the bite of an infected Aedes species mosquito. Looking at the visual diagram, we can notice that it is similar to its flavivirus family. It belongs to subtype 4, single-stranded RNA. It is enveloped, spherical, and has viron, which is about 40 to 50 nanometers in diameter. It is also a non-segmented and positive sense single-stranded RNA molecule, indicating that it needs RNA polymerase to replicate itself, and its genome also serves as a messenger RNA. Positive sense RNA viruses are considered infectious since their genome can synthesize all required viral proteins directly with the host enzymes. In other words, Zika fits the bill for being infectious. Looking at the graphics, we can see that the viron consists of a single capsid and two membrane proteins such as protein M and protein E. The surface proteins are arranged in an icosahedral-like symmetry. As we can see, the entire flavivirus genome is translated into a single polyprotein. The structural genes are located at the 5' end of the flavivirus genome. As a result, the portions of the polyprotein containing the structural proteins are synthesized first with the greatest efficiency. As one would expect, the reproductive cycle of Zika virus is similar to other flaviviruses. First, the viron attaches to the host cell membrane receptors via the envelope protein. This induces viron endocytosis. Next, the virus membrane fuses with the endosomal membrane, and the single-strand RNA genome of the virus is released into the cytoplasm of the host cell. It is then translated into a polyprotein, which is subsequently cleaved to form all structural and non-structural proteins. In other words, in early phase, it recognizes the target cell, attaches and penetrates, and then releases its genome. In the late phase, replication takes place at the intracellular compartments in the endoplasmic reticulum, resulting in a double-stranded RNA genome. This double-stranded RNA genome is then transcribed, resulting in additional single-stranded RNA genomes. Assembly occurs within the endoplasmic reticulum, and the new virons are transported to the Golgi apparatus and then excreted into the intracellular space where the new virons can infect new host cells. So in other words, late phase replication and transcription takes place in the endoplasmic reticulum. After the assembly, it is transported and the viruses are released. This is where the new virons can now infect new host cells. Now let's visit the chain of events leading to the disease. Zika virus is transmitted to people primarily through the bite of an infected Aedes species mosquito such as Aedes aegypti and Aedes albopictus. These mosquitoes typically lay eggs in and near static water and prefer to bite people. Mosquitoes that spread Zika viruses are considered aggressive daytime biters. Mosquitoes become infected when they spread on a person already infected with the Zika virus. Infected mosquitoes can then spread the virus to other people through bites. Once the Zika virus enters the human dermis, it enters the cytoplasm of keratinocytes and dendritic cells through receptor-mediated endocytosis to allow for a viral replication in the host. An accurate incubation period for Zika virus disease is not known. However, it is likely to be a few to 10 days. As illustrated on the left, the diagram simplifies the pathogenesis of the Zika virus. As we can see, the pathogenesis of the virus starts with the infection of dendritic cells near the site of the mosquito injection, then spread to lymph nodes and eventually in the bloodstream. Flaviviruses generally replicate in the cytoplasm. However, in the recent study, Zika virus antigens have been found in the infected cell nuclei. So, once the host is infected, it may be transmitted sexually from the mother to the fetus. We know that the transmission of Zika virus is predominantly through the use of mosquitoes as vectors. However, recent studies have indicated that human-to-human contact is possible, which would make it the first time an insect-borne disease was passed via sexual contact. As one can expect, both humoral and cellular immunity are provoked through the transmission. Both immunities are important to the control of primary infection and the prevention of future infections with the flavivirus. Interestingly, replication of the flaviviruses produces a double-stranded RNA replicative intermediate that is a good inducer of interferon alpha and interferon beta. The interferon that is released into the bloodstream helps limit the replication of the virus. However, it also stimulates innate immune responses and causes the rapid onset of the flu-like symptoms, which is characterized as mild systemic disease. The Zika virus has a remarkable history. It appears to have changed in character while expanding its geographical range. The timeline map summarizes the spread of Zika infection country by country from the earliest discovery in 1947 to the latest information as of February 2016. Uganda's Zika forest is the birthplace of the Zika virus, hence the name Zika. When first identified, the virus was only proven to infect monkeys. Even in the subsequent decades, when a dozen or so isolated human cases began to emerge, the symptoms were mild and Zika was never seen as a threat. But all of that changed in 2007 when the first large outbreak of Zika was reported on the Yap Islands in Micronesia. Many scientists believe that the 2007 strain of Zika had mutated from the original virus found in Uganda with increased virulence. Subsequent years saw the virus spread quickly through the Pacific Islands before landing in South America and Brazil in 2014, where there is a suspected correlation to an increase in the birth defect, microcephaly, and other serious conditions. Human Zika virus infection appears to have changed in character while expanding its geographical range. The change is from an endemic, mosquito-borne infection causing mild illness across Africa and Asia to an infection causing outbreaks from 2013 onwards. Outbreaks linked with neurological disorders including the Guillain-Barré syndrome and microcephaly. It is estimated that beyond the range of mosquitoes, infection has been and will continue to be carried widely by international travel. Needless to say, the United Nations Health Agency statement of Zika virus spreading explosively and could be a major risk to public health has led to Zika fear, especially among the travelers. Zika virus causes a disease known as Zika fever, which is characterized by a mild headache, macropapular rash, fever, joint pain, pink eye, and malaise. It is estimated that 80% of the people infected with the Zika virus may never develop signs or symptoms. For those that do, usually have mild, rarely requiring hospitalization. However, Since the outbreak in Brazil in 2015, there's a growing concern about the association between Zika virus infection in pregnant women and newborns with microcephaly and other severe neurodevelopmental birth defects. In recent studies, the most dangerous time is thought to be during the first trimester of pregnancy. With Zika's appearance on the map across the globe, mosquito-driven transmission cycle, and possible spread via sexual contact make Zika virus an important emerging pathogen whose global impact is yet to be discovered. At present, it is not clear and more research is needed to examine how the virus enters the placenta and damages the growing brain of the fetus. Additionally, an increase in Guillain-Barré syndrome has been observed in areas where a Zika epidemic has been documented. The disease affects person's own immune system, resulting in damaged nerve cells, causing weakness and paralysis. New studies have also demonstrated that prior infection with dengue or genetic factors may contribute to or increase cases of Guillain-Barré syndrome. Studies are ongoing to better establish the relationship between Zika and Guillain-Barré syndrome. Unfortunately, at present time there is no specific medicine to treat Zika infections. Treatment for everyone, including pregnant women, is directed at alleviating symptoms. Patients are advised to get plenty of rest and stay hydrated, may take acetaminophen to reduce fever and pain. However, patients should not take aspirin and other non-steroidal anti-inflammatory drugs until dengue can be ruled out to reduce the risk of bleeding. Patients should see their healthcare provider, especially if pregnant, and develop symptoms within 2 weeks after traveling to a place where Zika virus has been reported. No vaccine currently exists to prevent Zika virus disease. The easiest means of preventing the spread of the Zika virus is elimination of its vector and breeding grounds. Following the common sense steps offer better preventative measures. Wearing full-length clothes that are light-colored, using and reapplying insect repellent with DEET or Picaridin, using physical barriers such as screens, closed doors, windows, and sleeping under mosquito nets, and last but not least, cleaning and covering containers that can hold water such as buckets. With ongoing research and new information, it is important that healthcare providers are informed through CDC and Department of Health on current developments. Education should include recommendations for screening, testing, and management of disease, especially for pregnant women and women of reproductive age. Both CDC and DOH are making efforts for public awareness through social media and posters. CDC has issued a travel alert and has advised women who are pregnant or trying to become pregnant to consider postponing travel or take precautions to avoid mosquito bites. CDC also recommends to use condoms during sexual activity as a precaution for people with Zika virus concerns. Testing is critical to confirm the etiology of the disease, especially during the first 7 to 10 days of onset of illness. Viral RNA can be identified in serum, and real-time PCR should be conducted. Diagnosis for Zika virus infection includes PCR tests to detect viral RNA, as well as additional tests to detect Zika virus antibody in serum. IgM ELISA for Zika virus is detectable around 3 to 5 days after infection, but cross-reactivity with closely related flavivirus is possible. Plaque reduction neutralization tests can be performed to measure virus-specific antibodies. The CDC is currently working with a manufacturer to distribute assays to support laboratory responses to Zika. Avoiding bites of Aedes mosquitoes is a challenging feat. Oxitec Corporation has started the Friendly Aedes aegypti project to control the mosquito population. Pharmaceutical companies like GeneOne and Innovo have started making DNA-based Zika virus vaccines. This vaccine will be based on dengue vaccine, Dengvaxia, and is expected to be tested in humans later this year.